Okay, so thank you. Today we are very honored for the opportunity of having Pieter Visscher. He's professor at the Marine Sciences at the University of Connecticut and very interested in microbial mass, especially stromatolites. So this microbial ecosystem dominated life for 3.5 billion years, and so long before oxygenic photosynthesis. And the question arises: how stromatolites formed during their first 1.5 billion years on the dominated years and perhaps in space. So according to the work of Pieter and his colleagues, the arsenic cycle seems a good candidate. So welcome, Pieter.
Thank you, Laurent. Thank you everybody for coming. Wonderful to be in a very historic place with a lot of character. And I just had the pleasure, actually, and for those of you who haven't done this yet, in the museum to stand to serve on the back of a trilobite that I used to serve in Hawaii, so that was really great. But I saw the stromatolites by virtue of that reality, that was really great. So I will give a little bit in 2 slides an abbreviated version of what I saw there. And so end of the Vidéo, beginning of the Archean, the planet was in big disarray, and I'm talking to an audience that knows this very well, so forgive my ignorance as a marine scientist when it comes to geology. So, well, then we leave the Earth and just the impact of Gaia, and the moon is very close, which is important because of the big tides and throw up a lot of warm fluids at the site of the oceans that formed about 4 billion years ago. And when you go to the Museum of Natural History in Washington, this is the backdrop of the display. And this is sort of an idea how in the late Eo, early Meso-Archean, some 3.5 billion years or so ago, used this wonderful laser pointer. You see here the formation of these carbonate reefs. And of course, this is an artist's rendering, and the artist went to Shark Bay to get his ideas. And unlike what we just saw in the virtual reality next door, if the artist would have stuck his head under the water, you see that actually it's something much smaller and much more abundant than what was shown in that slide. But of course, the idea is the Earth has a lot of UV at the surface, with the less luminosity of the sun, lots of CO2, and hardly any oxygen, but virtually no oxygen in the atmosphere. So that's when we see the earliest evidence of life, and that's of course a little bit disputed when that first evidence is there, but this is the Warrawoona, about 100 kilometers west of Marble Bar in Pilbara in Western Australia, where you see some diversity in stromatolite structures, these layered rocks that you see there, which could be evidence that these are biogenic, but again, the jury is out on that. And so we have to investigate this further. Other examples, of course, in South Africa, Barberton, also beautiful, very large formations of these stromatolites. So when did it start? And we may see some publications coming out of that oxygen may arise a little bit sooner that I heard about this morning. But this is Alan Hutman's paper that goes back a few years, where he published that that he found stromatolites in Greenland, and based on the morphology there, you can see here he outlined it over there, and get some geochemical evidence like the rare elements, European lithium peaks there, maybe indicative of life, but not so fast as Abigail Allwood. And she wrote a rebuttal on this and said, well, if you actually you can look at this, but if you look around the corner, it looks laminated, and maybe these are ripples. And this is still going back and forth, and the jury is out if these 3.7 billion years stromatolites are the oldest, or 3.5 billion years. But we just think that at least in the Warrawoona, we have good evidence of the earliest stromatolites. So why is that important? Well, it's important because, of course, you can see the oxygen curve in red, going back to 4.5 billion years in present time. This is on a large scale. There may have been some whips of oxygen early on, but the great oxygenation event 2.5 billion years ago occurred due to the photosynthetic activity of cyanobacteria, but this is not a linear scale. And so on 30 to 40% of the time that there was this life, this based on the stromatolitic evidence, there was no oxygen or very, very little oxygen. And so the question is then, what did these ecosystems work on? So in order to think about that, we have to do a few things. And these are reconstructions of the oceanic composition, the main oceanic compositions. If you look at the middle one by one Canfield, you see that that going back in time, the earliest oceans were first, of course, rich in iron 2. Then with the onset of oxygen production, the sulfide was oxidized to sulfate, and we get that boring billion, which I think is actually very fascinating. When you have sulfur cycling and you have an oceanic ocean, the bottom waters are anoxic, the surface waters are oxic, and then it starts to go back maybe to an iron ocean during some of these ice ages, then finally coming to an oxygenated ocean, and that's what we see today. So why is this important? Well, the geological carbon cycle is really fueled by this little engine, this little pacemaker that we call the biological carbon cycle, where you go from CO2 to organic carbon and back again. And that is important because it connects to the atmosphere and the lithosphere, of course. Now, the importance of this is that when you do this carbon cycle in a very fast way, as biology does it, you need to couple it to another element cycle. You need two element cycles that are coupled to make geochemistry happen because it's based on redox reactions. And so today, of course, the bulk of the photosynthesis is oxygenic. The most abundant protein on our planet is Vidéo. And so that uses water to make oxygen. That oxygen is then used in respiration and goes back to the body. Right? So that is sort of the idea of how the biological carbon cycle works today. But then when you go back to the period where there is no oxygen, you have to rule the oxygenic photosynthesis out, and you have to anaerobic respiration as an important process. So then you have to start looking at alternatives. And so obviously, anoxygenic photosynthesis may have been around that uses the same process using light, an electron donor. In this case, it's hydrogen sulfide, and you make sulfate. The sulfate is respired, and now you have a combination of a carbon and a sulfur cycle. Important to notice is that cyanobacteria often, and this is an important thing to realize, that are very important in the oxygenic photosynthesis. But cyanobacteria can also photosynthesize without oxygen. They can also use some sulfur compounds, for instance, to perform photosynthesis. And so when we think of cyanobacteria and the onset of oxygen production, they may have been decoupled in geologic time. It could have been that for tens to hundreds of millions of years, these cyanobacteria, the earliest cyanobacteria, actually performed anoxygenic photosynthesis. Who knows? Okay, so we have to look at alternatives, anaerobic processes to make this carbon cycle fuel. And so that is important because obviously when we believe that the production of CO2 to organic carbon is mainly fueled on our planet, at least today, and maybe also in part of the Archean. Of course, that is based on photosynthesis, for that you need light, which means that this carbon cycle is somewhat decoupled in time. Right? The top goes only during daytime, and the bottom, if there's sufficient organic carbon, may go on during 24 hours. So, in short, there could be a decoupling of the two parts of the carbon cycle, which results in sometimes larger inorganic or organic carbon reservoirs, which can then to the atmosphere or can impact the lithosphere. In case of the CO2, of course, can carbonate precipitation in the direction of the. Okay, so knowing that, you can go and look at a few of the papers, and you have to realize, and I think most of you know, that when you go back in time, there's an enormous amount of arm waving, right? There's a lot of stuff that we don't know. It's somewhat speculative. So I'm using as an example two papers, the Stuken's work and Ward's work. Ward has done a recapture of the potential electron donors for photosynthesis that may have existed through time. And in this paper, Pieter T, they look at electron acceptors, so the respiratory part of the carbon cycle. And so you go back to 4 billion years, and they reconstruct it until present day. And here you see on the log scale the concentration of some of the possible electron acceptors, iron-3, sulfate, oxygen, and manganese are the ones that they chose in their paper. And so when you start thinking of that, in this period of time that we're now talking about, right, for earlier slides, what could that imply? Of course, today, photosynthesis makes oxygen, respiration uses oxygen, but that is not occurring until somewhere here, right, 500 years ago or so. So, iron, of course, the iron oceans were there during the early Archean, and so iron-2 is present for photosynthesis. You can use iron-2 as an electron donor for photosynthesis, but as soon as there's some oxygen, or also photochemically, you can oxidize the iron-3 very quickly, especially if the microbial life is not really ramped up yet, and iron-3 is not available once it precipitates as a mineral, so the jury is out on how important iron was. It could have been important, but for now, let's just rule that out. Manganese, very, very low in concentration early on, sort of Francois, that we can maybe revise this, but very low, so maybe not so important. But, and of course, the sulfur cycle also may not have started until a little bit later. But Schifru published some work and did some estimates of arsenic in the early rock record, and it is sort of in a range that it may be possible to support well. So that is our starting point, and hopefully we can find the electron donors that would, because you need the cycles, iron-2 produce sulfur compounds, nitride, hydrogen, arsenic-3 is possible as an electron donor for photosynthesis, and possibly with bacterial chlorophyll T, manganese carbonate as well. Okay, so now we're trying to start connecting this with some of the modern metabolites that I've been studying. This is in the Bahamas. This is a difficult place to work because it's in a ripping channel. That's the only reason that these metabolites exist there, because there's no opportunity for the eukaryotes to use the metabolites as hold fasts or to graze on it. It's really, so you can only brick dive over this, but there are other sites in the Bahamas that are a little bit more accessible. Now, when we study stromatolite, there's a strong connection with microbial mats. And this is microbial mat in cross-section. And what you see in this microbial mat are some layers of precipitation of carbonates and some storm events and the binding of trapping of carbonate minerals. Wolfgang Trumbine in the 1970s was the first one who really said, well, stromatolites and microbial mats are the same because they're both laminated systems. Well, here you have a lot of organic material, and stromatolites don't have that. So, how do microbial mats transform from these organic-rich sediments to liquefied structures that we see in stromatolites? Well, Wolfgang Trumbine was right and he was wrong, as much as I love him, and he was one of my mentors, because the lamination in the microbial mat is very different than the lamination in the stromatolites, and that would be a very different talk. But there are some ideas. Basically, one layer in a stromatolite, we believe, is actually an entire active mat with different layers in it. So the layers are not quite the same. It is called a microbial mat because you can actually lift it up and flap it over. It really stabilizes the sediments. Here is a blueprint for scale, so you can actually pick it up and flap it over. In this case, you see the cyanobacteria at the surface, and underneath there, you see the purple sulfur bacteria that photosynthesize and use light that penetrates a little bit deeper into the sediment and perform anoxigenic photosynthesis. Okay, so I'm quickly going to review a few of the processes that lead to the formation of calcium carbonate minerals, or are conducive to the formation of calcium carbonate minerals. So the first one is the populations of the microbes. And if you look at the stromatolites that you see in the Bahamas, of course, you have photosynthesis, and that photosynthesis takes place in about a millimeter thickness, or even less. And the activity, because it's very important not only who's there, but how active they are, the activity of the cyanobacteria in 1 millimeter is more than you see in a rainforest from the floor to the canopy. So that's about 30 meters or so. So activities are very high. As a result, you can get limitation of CO2 that is used in photosynthetic reaction, in which case, in the marine environment, you would deliver that by dissociating bicarbonate, the CO2 is immediately reduced by the cyanobacteria, and you produce OH minus, so pH goes up. The pH goes up, it means that calcium carbonate can precipitate, and so if you combine the two reactions on paper, then you would see that fixation of CO2 by cyanobacteria will result in the precipitation of calcium carbonate, and of course you can see that in this particular culture where it happens profusely. And you can play the game with a lot of different organisms that you see in the microbial matter. I'm not going to go through all of them. Just the light boxes are metabolisms during the day, and the night that are conducive of calcium carbonate precipitation that create conditions that may help the precipitation of calcium carbonate. The other ones, the darker boxes, are processes that do the opposite. They will dissolve the calcium carbonate. So it's really a balance of all these processes that make, in the end, produce the layer of calcium carbonate in that made layer from the stromatolite and organic matter disappears. Second component, very important, is the slime. We refer to them as exopolymeric substances. These exopolymeric substances, you see here an environmental SEM image of the Bahamian stromatolite there. You see this is a little bit more accessible, it's just in maybe half a meter of water. And you see cryo-SEM, this layer of SEM at the surface, and some focal image here, the green is Alan Decco's image, where you see the exopolysaccharides or substances, and in red you see the cyanobacteria, and in blue this is an oolith, one of these grains, and you see cells of some boring going on in the lipid activity. So this EPS, a lot of polymers, sugars, proteins, even DNA and other constituents, they have functional groups that can be negatively charged depending on the pH. And the carboxyl groups can be protonated easily, phosphate, and then sulfonate, sulfuric acids, what have you, and amino groups. And just for reference, here is where you will see later an arsenic. I'll come back to that momentarily. So the idea is that the cyanobacteria and other organisms in the mat produce this slime. The slime has these functional groups. pH goes up, they get deprotonated, and metal ions can bind to the slime later. So initially, it's like a dry sponge, put under the water, the calcium is the water, the sponge absorbs it, and no water gets through, until you get saturation of the binding sites. Then calcium is available. You may get a alteration of the matrix that can result in the template that can result in precipitation. But maybe the most important process is that this EPS is organic stuff, and bacteria eat organic stuff. And so when you degrade this, after it binds calcium, you release the calcium, and the organic material is respired to inorganic carbon. You get locally enhancement of the saturation index and you precipitate calcium carbonate. And this was Ducrot. I did an experiment in the Bahamas. When we did the transplant experiment, this was a mat that did not liquefy from a lake, and we transplanted it to an area where there was precipitation going on because of slightly different geochemical conditions. And so this is the beginning of the experiment, cryo-SEM. A few months later, we see cyanobacteria, but you see the EPS, the honeycomb structure is filling in the calcium carbonate, precipitated. You come back a few months later, and you see even more precipitation taking place. And that precipitation, we find, takes place on the nodes of this EPS matrix as a nucleation site for its precipitation. Third ingredient is chemical communication. Bacteria talk to each other. And that conversation is actually incredibly important, and it happens that these very small molecules, they're much simpler to make than complex larger molecules. And so the acylated homoserine lactones are very common communication molecules. And there's some other categories, and I'm sure there will be others that will be discovered. So the way this works is that you have a bacterium, and for some reason, it starts to produce, it senses a cue, and it starts to produce these homoserine lactones or other communication molecules. They are excreted to the environment, and the word form sensing means that you have to have a high density of these cells. And then the other of the same species, so this is now another of the same species, would take this off and you find complexation with a protein, you get DNA expression, and you get physiological change. So that's how they can talk to each other. But it's typically forgotten because there's a lot of work done in the medical world on this, that these compounds get modified in the environment. And we had an idea of that donné that actually bacteria can make change of their environment by secreting different chain lengths of this acylated homocysteine electron. But more important for us today is that there's also cross-talk, not only within the same species, but some of these compounds also affect other organisms. So now we have the material that the sulfate reducers excrete from the environment, and you can synthesize it in the lab. And when you do that, you can add it to another type of bacteria. This is a sulfide oxidizing bacteria. So the sulfide oxidizing bacteria gets its energy from oxidizing hydrogen sulfide with oxygen and makes sulfate. And this is a process that gives so much energy, about 800 kilojoules per mole, so you can fix CO2. So these are other trophic bacteria. This is what you find in hydrochromal vents, for instance. But you also find it in microbial vents. So at time zero, this soup of these bacteria, we give a shot of hydrogen sulfide. There's no sulfide here. We give a shot of hydrogen sulfide, and we can see over time that it's being consumed, right? Until a very low level, within an hour, it's gone. So if we do that in the presence of this communication molecule, you see that the consumption is different. And of course, the slope gives you an idea about the rate. So when the sulfate reducer sends out its signal, the sulfide oxidizer picks it up, it starts to metabolize faster. And if you do that at a lower oxygen concentration, it appears at first that the rates are similar. But then when you look at the product of the organism that sees the communication molecule, actually it has a different pathway, or a more complete pathway, and oxidizes it all the way to sulfate, which means it gets 4 times more energy. So the bottom line of that is that there is a benefit to this. So here you have a modern microbial map with cyanobacteria that make oxygen, which sulfate reducers at very high concentrations are not very happy about. The sulfate reducers sense that and they start talking to each other. But they start also talking to the sulfide oxidizing bacteria. Now they ramp up their metabolism, removing the oxygen, removing the sulfide that the sulfate reducers produce in their metabolism, which is inhibitory to some of the cyanobacteria. So now you have a system where you have cyanobacteria and sulfate reducers and sulfide oxidizers that typically would not exist in each other's company based on their physiological properties. Now they can live together and work at the highest possible rate that we can measure of individuals under the perfect conditions. So this is very important because that's what happens in a microbial map, and that makes metabolism that at first think incompatible, make it compatible. Last one, viruses. There will be a whole talk by itself, and I will just very brief about that. Viruses have proteinaceous capsids. These capsids can just work like EPS. They can precipitate calcium carbonate, and we can see that. Any of you have kidney stones or gallbladder stones, that could very well be because the presence of virus. It's not a viral disease, but the virus is present. And there are other mechanisms going to play at this point, but they can get trapped in the slime or they can infect the host. All of these things have an effect on the calcium carbonate precipitation at the end because the geochemistry changes. And that's all I'm going to say about this. I'll be happy to talk about it some other time. But now I want to come to my talk. And that is to link the extinct with extant microbial lives and see if we can learn anything from, if we can make a connection between. So first we start out in Western Australia, in the Pilbara. This area here is Pundiana, and where you find a wide diversity of different stromatolites. You see nice domal ones. These are right from the top. You see a lot of different morphologies there. Reconstruction of Tumbiana, the shallow lake, volcanic activity, maybe some geysers as well. And as I mentioned, you can find these outcrops with really a beautiful variety of different morphologies of stromatolites. And Pieter T's work there is ongoing, and you can see again club-shaped ones and small ones and big ones, and you can see here, if you go around the corner, that Abigail Elwood mentioned to Lutman, we have to do that in order to see that these are truly stromatolites. Okay, so a group from the IPGP, Pascal Philippot, was looking for nickel in this area because nickel is important in hydrogen metabolism, and he did some real coal, of course, that he took to Syncrophone Soleil, and he used the nanoscopium to make element maps. We later followed up, and I'll show some evidence of that momentarily. We combined nanoscopium with Lucia to also incorporate the sulfur maps. So Maris Forna, PhD student at the IPGP, published a paper some 8-9 years ago where they looked at the calcium maps on a very small scale and combined it with other elements, and I've left out the other ones, but the idea was that where you see in this hot map, heat map, you see enhanced calcium, you also see arsenic, and Kevin Lepeau actually showed that the organic globules in there were associated with arsenic, and the sulfide was associated with the iron. So this was not a smoking gun, but some ideas that perhaps arsenic played a role in this particular stromatolite from 2.7 billion years ago. So at the time, Pascal Philippot and I crossed paths, and I was on the quest to find microbial maps that were completely anoxic. And so those are hard to find because there's oxygen in our atmosphere today. So I set off with some colleagues from Argentina, and we looked at a variety of lakes in the high Andes. This is on the Argentinian side. See, there are no eukaryotes here, so that is a good sign. This may be compatible with early life conditions. Of course, UV is very high there. We crossed into northern Chile, and outlined here is the Atacama Desert, where, of course, the lithium of your electric car come from, because it's high-grade lithium, and a lot of mining activity. And a colleague of ours had an environmental company in Chile and monitors different sites, and so we had access to the lakes there. But this was the early image that I showed you of the Smithsonian, and when we found this lake, La Brava, it was very similar. We have volcanoes there, we have shallow environments, we have high UV, and we have a wide variety of different microbial structures. But in the lake itself, there are cyanobacteria in these structures. So this is not quite right yet, but since the humidity is less than 1% in Atacama, there is a lot of evaporation of the water, and so the water has to come from somewhere. So we started to walk around, and we found, so this is our field site, we found this red river, which led us to a source, a spring that fed, it was one of the springs that fed from groundwater, volcanic groundwater, that is feeding La Brava, the lake. So this is the lake, and the field site is here, and you can see the source is here, the spring. And so halfway, the spring, all along the river, we did some, or the stream, we did some measurements, but we picked one in the middle, and you see beautiful red structures, and these are purple salt bacteria. Right? So these are anoxigenic coated bacteria. Now the spring is very high in hydrogen sulfide, so we did that momentarily. So if you take a piece of the map, you see this layer of purple salt bacteria, which is almost a pure culture. And the species here is Ectothiorodus spider, which means that it's sulfur outside the cell, but this is one of the species that can use arsenic-3 as electron donor for photosynthesis. Okay, so we are at high altitude, 2,500 meters or so, high UV, low humidity, pH of the stream fluctuates around 8. Very interestingly, the diversity is almost 3/4 archaeal, which is quite unusual. Sulfide at the source fluctuates a little bit through the year, but is up to 2 millimolars. The arsenic is 1.5 millimolars, so really high in arsenic. No chlorophyll A in the system, but we find a bunch of other photopigments. We find aragonite and calcite, but there's also some arsenic present, not in by XRD, but in when we do SEM with EDS. And lithium almost tenfold. Okay, so we go there and we do our measurements with microelectrodes and other things, and this is sort of the picture that emerges. The overlying water, of course, you get diffusion of oxygen into the water, but it flows fast enough that it is a filter, and oxygen never reaches the microbiome itself. We went out in 4 different seasons. In the end, I actually also used optodes to make sure that I couldn't see any oxygen, and oxygen is never present in the microbial mat. This is the microbial mat. This is the top centimeter you see here in cross section. And so sulfide enters the mat, and is also produced by sulfate reducers deeper in the mat. And you see a minimum of the sulfide at about 2 millimeters where you have the purple layer. So this is photosynthesis using hydrogen sulfide. This is a map of the sulfate reducing activity. The pixels that show where you find activity. This is not so important for my story right now. But we took a little sliver here and went to synchrotron, and we looked in more detail at 2 points where we used addition to these X-ray fluorescence also xenos. And these are 2 standards that we see in these 2 points, 2 peaks. Now, why is that important? 2 peaks means that you have arsenic 3 and arsenic 5. And if you have 2 oxidation states, you can make an element cycle. So now we have a carbon cycle and an arsenic cycle combined, what's the potential of that at least, to make the system work. When we do similar experiments, and I showed you this before, we have a mat with no arsenite in it because it's pre-incubated, and we gave it a shot of arsenite, so arsenic 3. In the light, you see consumption. Under other conditions, we don't. When we do the same experiment with arsenic 5, which can respire, we see that nothing happens unless you add organic compounds like lactate and acetate. Then you get consumption of that compound over time. So, summarize of our first measurements, we see that there is oxidation of arsenite, so photosynthesis on arsenite and on sulfide. Later observations have shown that these bars can be about equal, but in this case, you could get more sulfidic photosynthesis, but the respiration is the same for sulfur and for arsenic. Okay, so genes for arsenic cycle are there, and so you can, using the genes, you can make a complete arsenic cycle. That all looks good, but often I get the question, where is the arsenic, other than being recycled? And so these are some images from a different map, but very similar to what we believe is the case in La Brava. And this again are synchrotron maps, and in red, this is a better image to look at. In red, we see arsenic. And what you see here is a veil of EPS. So the EPS is very high in arsenic. And actually, when we go to a sulfidic environment, we see that the functional groups are predominantly sulfidic. So maybe because of the high presence of arsenic in this case, it's being incorporated in that slime that the organisms produce. This is a map, and in yellow you see the calcium, and you see some red, especially in this layer that it's precipitating. So there's a combination of calcium and arsenic as well. Well, it is known from other studies that arsenic 5 preferentially absorbs to carbonates, and arsenic 3 does not. So arsenopyrite, maybe a little bit of that, because this would be a little pyrite inside this fluid. But most of the arsenic is present in EPS, and then later in the form of calcium carbonate combined with calcium arsenate. So the same story again here. The functional groups, calcium binds to the functional groups, and then either arsenate attaches to that and precipitated this carbonate arsenate species, or alternatively, arsenate is incorporated in the EPS and binds calcium, and you get precipitation of the carbonate arsenate complex. In the case, the result is the same. So coming to the end, we can rule out oxygenic photosynthesis in our system. We have, of course, no aerobic respiration. And nitrogen is very low and mainly used for biomass, so nitrification not so important. Manganese we don't measure in our system. There is some iron, but not too much either. And so arsenic respiration and sulfate respiration are important. Now, going back in time, one thing that is very important in ecosystems is that, of course, the more energy you get out of a reaction, the more favorable it is. So when you have arsenic and you respire that, you get about 20% more energy out of the reaction than when you use sulfur. So that could be an indication that, that arsenic respiration is favored over sulfate respiration and both are present. So perhaps then you can think of a system where sulfur and arsenic cycling coincide, where arsenic cycling is more important. Now, one last thing is precipitation of calcium carbonate under different conditions. In the blue reactions are some metabolisms, respiration metabolisms using sulfate, and in red are the same substrates being used while respiring arsenic. And this is the table to look at. When you see the change in the pH, negative sign means the pH goes up, so more alkaline conditions, so favors calcium carbonate precipitation. You see that the red reactions that use arsenic are much more favorable than those using sulfur. So in other words, in arsenic cycling, in principle, on paper, you can precipitate more calcium carbonate than with sulfate. So then, in the end, perhaps coming together, we have hydrothermal processes, we have weathering and volcanism, producing pools of arsenic-free and reduced sulfur. And so, side by side, you may have an arsenic cycle, photosynthesis of an arsenite, respiration of an arsenate, and then producing arsenite again, so an arsenic and a carbon cycle, but also a sulfur cycle where hydrogen sulfide is the donor for photosynthesis, and sulfate that is produced is being respired. So a combination of an arsenic and a sulfur cycle combining with the carbon cycle, and of course there is also the activity between the arsenic and the sulfur cycle, but that's getting into too much detail, but that happens as well. So then perhaps Ljubljana, maybe it looks something like this, is another site in the Atacama where you have fields of formaldehyde. So with that, I invite you to ask any questions you have. Thank you for your attention.
Thank you very much. I was wondering, both in your detailed observations to the lights, and maybe in the literature about the early Earth, sulfur zero can also be an electron acceptor.
And donor available, yeah.
Yeah, and I was wondering, both in the very specific case of the microbialites, or the system you looked at, and maybe also for the big picture, it's there, right?
Absolutely.
And in organic phases sometimes.
Absolutely.
And I was wondering the relative importance when compared to arsenic, for instance.
Yeah. So that's a very, very good point. You can envision a shorter sulfur cycle between hydrogen sulfide element and sulfur, and that definitely must have existed. When we look at the analog, which may not be the perfect analog, we see that that they both play a very important role, and it's about 50-50, but it would really depend on how much sulfur and how much arsenic you put into the system. So your point very well taken in that boring billion, for instance. Obviously, that's going to be the sulfur cycle is incredibly important and probably even more.
Do you have evidence in your science of sulfur zero reducers, for instance?
We haven't tried for that yet. That would be a good point. There are also, of course, in addition to the reducers, you have oxidizers, but you also have the disproportionation that reduces the fermentation, which is a process that we don't often, at least I don't often observe in microbials. It does exist, but it's never as dominant as it may be in certain marine environments, as the oxidized people. That's a good one. So should look at that.
Iron, okay, in the minaire, there's significant amount of iron. And I'm always surprised to see that the arsenic is not there. So there's little actually, because there's so much work on the affinity between the fractionation, the partitioning of the arsenic range, the iron sulfide and so on. So I'm just wondering, so does it mean that basically it was pyrite or not forming at the same time when the arsenic is there? Because yeah, that's surprising that they would not compress. Arsenopyrite is supposed to be an arsenic rich.
Yes.
So unless you have like a difficult pyrite forming somewhere else, I'm always surprised by that. I'm just wondering how it works. I mean,
No, yeah, and I cannot really answer that question. One thing that definitely needs attention is also the interaction between the arsenic and the sulfur cycle in different forms than the precipitation with iron and the arsenopyrite. We do see some arsenopyrites in the solar system as well, but it's mainly in the carbonates that we find it in this modern system. That does not explain everything that you're right, that we find it in Guyana. Of course, what you see in the rock record is an integration over long periods of time, and this is so there's not a compatibility in that respect. But that's something to consider for sure, yeah. And then we figure out what happens with that, and maybe the input of arsenic was much lower than in the Guyana system for sure. Yeah, that's a very good point.
Okay.
how much arsenic do you have in the groundwater?
So when it comes up, it's 1 millimolar or higher.
Okay.
Yeah.
So it's pretty high.
So we see that.
Okay. And then the fluxes, so right now, do you have any clue about the rate of the transformation, let's say between the sulfide to sulfate or and arsenite to arsenate?
I should be able to calculate that from our slurry experiments where we have some rate measurements, and we have measurements of course of sulfide and arsenic across 250 meters of the stream. So I should be able to calculate that. Yeah, that would be interesting.
Yeah, that was a bit my question. Do you have the hydrology of this lake? Is that a permanent lake? Does this concentration are constant over a year? Or?
No, the lake level changes a lot. In wintertime, it's much lighter than during summertime. So, but the flow rates that we measure from the spring are fairly constant throughout the year. Maybe 20% difference between summer and winter.
I have a question about the communicators. We will talk about the communicators between different cells, things like that. But later I saw the photos like in the lakes, there are several different microbiologists, bacteria, and then they have different colors like the pink or things like, why don't you focus on the pigments? Because I think pigments are also very important for different bacteria and for the concentration or the abundance of the communicators you mentioned before.
So the concentration of the communication molecules, the chromosenting compounds, are not easy to measure because they are modified by the chemical environment. Right? One of the important things, sort of, is not a direct answer to your question, that to consider is that the presence of an organism is very important to demonstrate. Right? And that's what you can do with molecular diversity studies. You can look at the presence of RNA, which may tell you something about a process, but sometimes RNA is turned over very quickly, and it's actually very difficult to get RNA out of mass. So you really also need to know the activity. Right? So activity measurements are not necessarily coupled to abundance. Of course, there's some correlation between the two. So yeah, pigments are important, and that's another way to look at abundance of specific groups of organisms. But you still need to know the activity. You can have a group of organisms sitting there and not doing a lot, while others in the same abundance will do. So yeah, the importance is also to know the exact, as much as you can, the activity of the organisms in addition to the abundance.
Do you know why is cyanobacteria not present in this?
They may not like the arsenic. They might definitely not. The sulfide is also very high, so I don't know.
Because in Laguna Negra, lake in Argentina, a lot of arsenic. And
It could also be that
they can stand it.
it could be that they're competitively excluded if you have high levels of sulfide and arsenite that can be used by the purple sulfur bacteria in a very efficient way. Maybe the cyanids don't get a foothold. And what I think what Vidéo showed is he did experiments with microcoleus and with thiocapsa and was looking at the anoxigenic photosynthesis he did not use hydrogen sulfur but thiosulfate, which is less deleterious to the cyanobacteria. And he found that the purple sulfur bacteria could outcompete the cyanobacteria very, very quickly. So I think it may be a competitive disadvantage that the cyanids have. Although, again, we don't know what kind of cyanobacteria were around early geologic time. So maybe those were very different and were more competitive than the purple sulfur bacteria. They must have been because they developed quite exclusively. So that's an interesting evolutionary question, actually.
Or maybe according to this project, what we have to touch space in from now, so we go to the chemical archive, so arsenic that is calmate, so
yeah, that would be cool. Let's bring some samples back from Mars and look at those. So I can think of some people that I would like to send to Mars to the study. They're in the collections right now in the United States. Something like that. But I think we have to think outside the box, maybe not specifically to arsenic only, but you can think of similar systems working on selenium. And as long as you have two oxidation states, I think it's a potential candidate to support carbon cycling. And so I think very, very right outside the box. And I think the pixel instrument on the current mission will help a lot with identifying other potential elements. Again, the issue is a little bit similar to the activity, though. The reservoir size does not necessarily indicate the importance of an element, right? So, if I would put it's almost lunchtime, so I put a bowl here of Brussels sprouts and one with very nice Belgian chocolates. The chocolates will go, and the Brussels sprouts leave. And if you see the reservoir is big, but it's not an important, right? So it's a similar thing with these.
Excuse me, just one point about that. There is still some colloidal iron oxide.
Yeah.
So this and probably the arsenite, they are just controlled like this. At some point, you probably need iron reducers to get.
Yeah, but we don't have any in this system. We don't have them, but. And then there's evidence of very early dissimilar iron reduction. Right? I think there's some studies in South Africa. I think it's exactly 6 or so, 7 maybe even. They, based on isotopic evidence, they claim that it's
here you are sure there are no because you.
Yeah. And then one of the big questions is 75%, 72 to 75% archaea. What are they doing? So I don't know. That's remaining busy. Yeah.
okay, we can teach our doing something after, but thank you.